Hi, good morning, everyone. Hope you are having a wonderful day. This is Tony. I'm here at the Bio Creation Conference with H3 Africa and H3 Bionet here in Cape Town. And here I have one of our participants with us for the conference. So tell us about yourself, sir.
Thank you. My name is Martin Nganga. I came all the way from Kenya for this the particular conference. So far, it's been interesting, very engaging, and I like everything about it. Thank you.
Okay.
Yes.
So Martin, you're from Kenya. What type of work do you do? Are you a data scientist? You're a bioinformatician?
Okay. I'm not a data scientist. I am a microbiologist, but I happen to deal with a lot of data. So here I'm presenting work on the curation of antimicrobial resistance genes from Africa. So the work came from the need for microbiologists to have a curated resource for antimicrobial resistance genes.
Okay. So you come from a very biological background.
Exactly.
And what type of, firstly, tell us what type of pathogens you work with. Is it viral, bacteria? And then secondly, tell us about your academia career. Do you have a lot of support in your institution, things like that?
Okay, thank you. I deal with microbial pathogens,
Okay.
bacteria to be
Bacteria.
specific. And as I said earlier, you realize along the way that you require a lot of data. So then that's where the interest on bioacquisition came from. So I'm not particularly a data scientist, but I'm evolving to
Oh, you want to?
become one.
Okay.
Yes.
So you sort of work in the laboratory, you collect the samples and then do the extraction and try to get the data. What type of equipment, how does your method of that work?
Yeah, in microbiology, we do mostly experimental work.
Okay.
You collect bacteria, you isolate bacteria, then you sequence the DNA,
Yes.
and that is when you get a lot of that genomics data.
Okay. And what do you use? ONT, Illumina,
Yes, we use Illumina sequencing.
Illumina sequence. So
Yes.
that's the short read.
Yeah, exactly.
Okay. And then you know what genes you're looking for.
Yeah,
So it's
but
very
mostly
targeted.
you do uh, for metagenomics, you sequence the whole community of
Okay.
microorganisms there, but we also do, you can also do a whole genome sequencing of a particular pathogen.
Okay, right. So I see you use Illumina, and then you obviously do either whole or targeted sequencing with
Yeah,
metagenomics. And when you
exactly.
isolate the interest gene, and
Yes.
that's now you're going to be doing essentially downstream analysis,
Yeah.
which is basically where the data scientist or the bioinformatician part comes in,
Yeah.
and you now wanting to essentially gain skills to
Yeah.
become a
Exactly.
proficient bioinformatician.
Yeah.
So that's obviously, either coding or running pipelines. And then obviously by attending this conference, you hope to learn some of those skills and
Yeah.
learn about the different standards.
Yeah. Because as I said, my background is in microbiology, experimental microbiology.
Yeah.
But then the skills on data handling, data
Yeah.
science, I didn't have them. I've
Yeah.
been learning them along the way through YouTube, through the resources available at the bioacquisition website.
Yeah.
Yeah, so this particular conference
It's going
is
to
very
help you
important.
facilitate. And
Exactly. And also I've been meeting people now from different background, data scientists, bioinformaticians. So it's a good opportunity to network and
yes, sir,
to see
definitely.
the opportunities for collaboration.
Yeah.
Yeah.
And then you obviously mentioned you come from Kenya. Tell us, about the city that you're in, type of support that you get from your institution.
Yeah, in terms of institutional support, I would not say it's there. Mostly, researchers, you are left to your own devices to
Yeah.
look for those resources.
Yes.
So I wouldn't say that institutional support is there because there is no dedicated data scientist you can go to.
Okay.
Or bioinformatician. So mostly you navigate yourself. Maybe that's something you can build into the future.
So essentially, you don't have a lot of support. There isn't much mentoring or resources that facilitate you, in Kenya. So as you mentioned, for that transition you need to occur, you need to sort of find, these skill gaps, find mentors, or find online resources that can help you to, build those skills.
Exactly, that's the gap I see. I think from the interaction, the workshops I have seen here, there has been that problem repeated across many other institutions in Africa.
Yeah, certainly.
So it's a gap that needs to be filled. And I believe like attending this conference is one of the ideas. I can take those ideas back to my institution.
Yeah.
Yes.
And then we obviously try to partner with the institutions like the African CDC, some of these big G4GH global networks that trying to support, you know, since the COVID pandemic, there's been really need to have skilled scientists, especially in the lab, and obviously for better pandemic preparedness.
Yes.
If you to give a few words to some of these big institutions like the Africa CDC, what do you hope that they can do to help you or other young scientists? Is it more training that you need? Is it more equipment that you need? Is it better type of workshops like this that can help you?
Yes, I think the kind of support that would be needed would be in terms of training,
Okay.
sustainable training.
Right.
Maybe, maybe at least once every year they can hold workshops like what we have seen here. That would really help. Provide some of those resources.
Yes.
And also partnering with research institutions. That I think that would really help a lot to build that capacity that is lacking.
Oh, I see what you mean. So because there's a lack of skill set on the continent, what you actually need is for institutions like the Africa CDC to run frequent workshops, to invite people from different labs, to have a field expert that can tell you how to use Illumina, how to use ONT beta, how to actually take your file and clean it up and get your gene and compare it to the database, teach you those skills, but also have repeat training so that you can continuously upgrade your skill set.
Yes, exactly. Not a once in a lifetime activity, but if that can be made sustainable, frequent, I think that skills gap that is there would be closed within a few years, maybe in 5 years.
No.
Going to the future.
Yeah, I think that's really helpful. It's a good insight that we can obviously take back to our partners and training institution for them to sort of think about.
Exactly.
And then in terms of, we are in the fourth industrial revolution. There's a lot of AI, artificial intelligence. What are your thoughts on that? Do you work with AI a lot?
AI, I would say, has been instrumental in my work because some of the skills I have gained in terms of programming, I don't think I would have gained them if there were no AI, because with AI, you can write a script. If it has errors, you can
ask for it to
ask
correct
the AI
you.
to look for the errors to
Yes.
correct. So it has really accelerated the learning curve. But at the same time, there are challenges with AI. As you know, AI hallucinations. So, but generally, the impact has been positive. Yes.
So you use the chatbot like ChatGPT, Claude on a weekly basis, and
Yeah.
you ask her to correct my code in Python or NetFlow.
Exactly.
Okay.
I use Cloud as
Claude. Okay.
very good features.
Yeah.
So, in doing that data analysis, data analytics, all that workflow programming,
Okay.
it's been
Very
useful. I
interesting.
think the skills what I'm able to do now without AI, I don't think I would be able to do.
No,
If I were running with the traditional methods.
yes. No, it's good to know that. So as we come to the end of this interview, Martin, I think the final question I would like to ask you is, what advice, you have been in the scientific field for a very long time, what time of challenges you experienced, personal growth that you've done, if you were to give some advice to someone, that's in high school, that's just coming out of university, that want to pursue science, tell them, your mistakes so that they don't make the same mistake, give them some recommendations.
If I were to advise somebody who is starting science, one of the things I would say is to focus on research integrity. That is one area that people ignore, then it catches up with you in the future.
Got you.
You do credible research, publish in
Good journals.
good journals, then
You'll be recognized.
the rest will follow.
And then what type of challenges have you overcome? Tell us about some of your problems.
For my kids, I think the biggest has been being able to get funding from projects. That has been one of the
difficulties,
major challenges so
yeah,
far, even today.
Yeah.
But I still overcome them. I apply for grants.
Yes, sir.
Sometimes you are successful, other times you are not.
Yeah, no, I understand.
Obviously, funding has been the major challenge so far.
No, completely understand. Thank you very much, Martin.
Thank you.